Thank you, Marcy. So hello, everybody. I am Claire, class of 23. And I'm very excited to be the one to kick off this STEM series. As a very recent graduate, I thought that maybe the best thing I would have to offer a room of Scripps students would be the perspective of someone who's still kind of figuring stuff out. So I'm going to talk about two of the most formative research projects I worked on during undergrad, which both sort of fall under the umbrella of high-throughput biomolecular engineering. So I'm going to talk a little bit about bioluminescent proteins, a little bit about DNA catalysts, and then I'm just going to generally touch on my journey and how I got to where I am today. So depending on who you are in the room, some of this stuff might go totally over your head, and to this table of people it might be incredibly obvious, so just bear with me. I'm hoping everyone walks away having learned something new. Does it work? Great. All right, so I'm going to talk just briefly about the most formative experiences I've had in science over the years. So I matriculated here at Scripps in fall of 2019. By the end of my sophomore year, I declared a biochemistry major, and then in the fall of my junior year, I joined Aaron McCont's research group at the Keck, where I began researching the enzyme firefly luciferase, and that project continued into the summer of 2022, where I worked on it full-time. The following summer, I spent at the University of Illinois, Urbana-Champaign, studying DNA science in the research group of Scott Silverman. And then in the fall of 2023, which was my final semester in college, I worked on two projects that were really inspiring to me, one of which was an independent study in which I wrote a digital primer on the topic of directed evolution for the ACS In Focus series, and that's something I'm really proud of. The other thing is that I began doing some research on XNA polymerases as part of class, but then I continued that research into this most recent summer as well. So today I am about 2 weeks away from starting my PhD. I'm on the chemical biology and molecular biosciences track at Caltech, where I have no idea what I'm going to study, but you can ask me again in a year and I might have a better answer. So today I'm just going to be talking about these two research experiences I had because they were the most formative in determining who I am as a scientist, but also because I just have the most to talk about with these two things because I spent the whole summer working on each of them. So starting off, I'm going to talk about firefly luciferase. This is the first project that I ever worked on in a research lab. Firefly luciferase is a bioluminescent enzyme. I show the structure over there on the right. It catalyzes this oxidative reaction of its substrate D-luciferin and produces light as a byproduct. So this is the chemical reaction and the protein responsible for making fireflies glow. Firefly luciferase isn't the only luciferase. There are also a number of beetle and marine luciferases, but we care about firefly luciferase because it has a lot of important implications in research and biotechnology. A really helpful application of firefly luciferase, or FLuc for short, is that we can take the FLuc gene, transform it into a bacteria like that E. coli, and then if we feed the E. coli enough of its substrate to luciferin, it will glow. And this is a really helpful property and used very commonly as a reporter in biological assays. It also has pretty cool biotechnological applications if we sort of scale up that same idea. By transforming the FLuc gene into, say, a particular cell line in a mouse, and then adding D-Luc, we can visualize that cell line over time in deep tissue. This is very exciting. You can imagine this technology being used for, say, tracking the growth and movement of a tumor over time in the human body. However, before we can use it for that kind of a purpose, we have a few main hurdles we need to get over. One of which is the light emission color of firefly luciferase. So firefly luciferase gives off a sort of green-yellow light at 560 nanometers, which is not great for in vivo imaging. There's this ideal range for in vivo imaging between like 700 and 900 nanometers. And the reason we care about that range is because everything outside of that range is going to interfere significantly with the absorption spectra of hemoglobin. And all that means is that if we put this firefly luciferase gene into a deep cell line, in like a human, for example, or anything lower than like skin level on the mouse, the majority of the light signal emitted by that luciferase is going to be absorbed by like the blood in the body, the hemoglobin and the oxyhemoglobin. So thankfully, we can overcome this problem by protein engineering. And all that means is that we can make specific mutations in the protein, in the firefly luciferase enzyme, that cause the light emission spectra to be more red, closer to this desired range. And this is what we call redshifting. And many people have done this. There are several different luciferase mutants that give off a more redshifted light color. However, despite this, we still don't really understand why this happens. There are a lot of different theories about the redshifting mechanism that exist, but no one has really been able to come to a conclusive decision on why that happens. So we can have redshift luciferases, but without knowing exactly why that's happening, it can be hard to predict what kinds of mutations might cause redshifting in the first place. So in order to make a redshifted firefly luciferase mutant by a traditional biochemistry approach, we would start by designing our mutant. And what this looks like is looking at the structure of a protein generally, choosing an amino acid residue that seems important for catalysis, and then mutating it. So we might design one mutant at a time, or a handful. We're going to clone that into a plasmid, that's just a circular DNA. We'll transform it into bacteria, say, express it in bacteria, and purify it out of bacteria to obtain your mutant luciferase enzyme, at which point we can conduct comprehensive kinetic assays to characterize that enzyme. In this case, we're thinking about redshifting. We want to know what color the light is, so we might conduct these luminescence spectroscopy measurements to look at the light emission profile of each mutant. Now, this is a very effective process. It's worked many times. However, it's quite slow. This whole process might take anywhere between 1 and 2 weeks, give or take a week, depending on things working well the first time, which they hardly ever do. And there are pros and cons to this kind of approach. The major pro to this kind of approach is that it is a hypothesis-driven approach. And as anyone who knows anything about the scientific method, hypotheses are the core of that method. We like to test hypotheses, state hypotheses, test them, and then determine whether or not our hypothesis was correct or not. It's a very good way to do science for a reason, and that is a traditional way to do things, and it's good. The other thing is that we're taking nice, rigorous measurements of our enzymes, which is very helpful. We're learning a lot about each enzyme we're making because we're just studying one thing at a time. However, we're only going through a handful of mutants at a time. This is what we call low throughput. And that means that although we're learning a lot about each individual enzyme we're making, we're not learning a lot overall about luciferase and, say, the mechanism of color shifting, which is what we care about. The other thing is that in order for this kind of process to work, it requires having extensive existing biochemical knowledge of the system. So if we knew, for example, how the redshifting mechanism happens, we could easily test hypotheses the traditional way and make changes that we think are going to cause redshifting, and this would probably work a lot better. But since I said before, we don't really know how that happens, trying to predict redshifting mutations is often a shot in the dark. So the alternative to this is taking a high throughput approach in which we're following the same kind of workflow, but instead of making one mutant at a time, we're going to make hundreds of mutants at a time. And this is what we call a library. We will assemble plasmids the same way, express our proteins the same way, but then instead of taking these rigorous measurements of each individual candidate, we're going to sort the diversity in our library by a screen. So if you can imagine here, this is a 96-well plate. In each of these wells, we would have a totally different luciferase mutant. Now, importantly, at this step in the process, we would not know the identities of any of these mutants. But for a second, we're going to decide not to care, because we can then work backwards with high-throughput sequencing to determine what we were looking at in the first place and assay the hits that we find most important. And this process can take about the same amount of time, with the caveat that high-throughput sequencing takes forever. But the great thing about this is that instead of working through, a handful of mutants at a time, we're going through hundreds at once. So this is a high-throughput approach, and although we're sacrificing the rigor of the measurements, we're learning more overall about the protein and how it works, because we're looking at how hundreds and hundreds of mutations affect the emission of color all at once. This is what we would call an outcome-driven approach, as opposed to a hypothesis-driven approach, where instead of making a hypothesis and testing it, we're stating our desired outcome, being a redshifted luciferase mutant, and screening hundreds of mutants all at once, and then eventually working backwards to figure out how we got from point A to point B. And the great thing about this kind of approach is that it does not require extensive background knowledge of your system, which is great when you're trying to figure out how something works in the first place. The con is, of course, that we're sacrificing the rigor of the measurements, although, of course, we can go back and re-assay the individual hits that we pull out of screens. And this also requires having high-throughput infrastructure, which not everyone has access to. But in our case, we do, and so this is the approach that we took. More specifically, this is what that workflow looked like. So we started with something called the statistical coupling analysis, which was a computational algorithm that identified 20 different amino acids on the luciferase protein that we mutated. We created 20 libraries at that positions, which mean 20 site saturation libraries, which means that at each of those 20 positions, we cycled through all 20 possible amino acids, which means we had 400 total mutants to sort through, which is a lot. And then we ran through 2 different plate-based screens. An activity screen first to separate out the active mutants from the inactive mutants, those which no longer produce light, we don't really care about. And then we're going to go through that color screen I was telling you about before to pull out the novel redshifting mutants that we care about. In order to identify the sequences that we're looking at in the first place, we can compare that pre-screen population, the active population, and the redshifted population via high-throughput sequencing, which is going to spit out this giant database of information that we can sort through and identify inactivating and redshifting mutations. At which point we can take those mutants which we're most interested in and do those rigorous measurements. So in this case, we did bioluminescence spectroscopy assays to look at the redshifted emission profiles of interesting mutants. So this is what I did in the summer of 22. This was my first research project ever, and I worked through all 20 of the libraries with the help of my partner, Nicole. And it was a really exciting process. It took about 9 weeks, and we learned a ton in the process, but I'm just going to walk through some interesting highlights. So this is what that color data looked like on the right here. And you can see down on the x-axis, those are all of the 20 luciferase libraries we looked at. Hold on, I gotta go over here. For reference, this is wild-type luciferase. So this is that green-yellow light that natural luciferase emits. Up here, this is COL2. This is a known redshifting mutant. So for comparison, the higher up you are on the color score axis, the redder your light is. And so each point on this graph represents a unique luciferase mutant. And you can see that there's a ton of variety in this data, but there are a few key patterns that are easy to point out. For example, you might notice that positions 229, 237, 286, 352, 372, and 431 display a lot of redshifting characteristics. It seems that these positions that changing the wild type residue to anything other than wild type appears to cause redshifting. That's very interesting. What's even more interesting is that when we plot those 5 enzymes onto the protein, they form a really nice pocket around one side of the active site. So that's these protein enzymes here, residues here. They're all forming a pocket around the phenolate of the substrate, that's this side of the oxoluciferin here. And that's very interesting. We really like to see our data translate from numbers to conclusive structural evidence. So this is very exciting and interesting for a reason that I will get back to. So remember this. I'll go back here. The other thing we did was we took 6 different redshifting mutants that we found in our experiments, and we took the bioluminescence spectroscopy of those mutants. And so that's what you're seeing here. That black line on top, that's the wild-type luciferase, that's that natural green-yellow light. And then all those others curves are the redshifting mutants. And what's interesting about this data is that where we might have expected redshifting to look like a gradual movement of that emission peak into the red, it actually seems to be a preferential stabilization of 2 different emitters that's causing this redshifting. So you can see that dashed green line there is pointing out the first hump in all of the curves, that's like the green emitter, and then the red line is showing that red emitter. And this isn't anything totally new. This is a pattern that people have pointed out before, but it's nice to be able to sort of have conclusive evidence agreeing with other theories. People have generally associated these 2 different emitters with the keto and enol form of oxidized luciferin. That's on this side of that substrate. You see this tautomerization that people have associated perhaps with being responsible for these green and red emitters. But what's interesting is that if you recall, our redshifting mutants did not cluster around the tautomer on this side of the substrate, but rather on the phenolate, which doesn't change at all through the entire oxidative reaction. And that's very interesting because not only is this telling us that there's a side of the enzyme that we haven't been looking at, but also that there's something important about the active site and specifically the solvent network that has been ignored previously. So chemically what this tells us is that there's some kind of important solvent-mediated downward stabilization of the green emitter, or perhaps a destabilization of the red emitter that is interrupted when we mutate residues on this side of the active site. But what I find interesting about this is just proof that taking an unbiased mutagenesis approach sometimes causes you to find things that you wouldn't initially look for. Because the phenolate side of the substrate isn't changing a lot in the reaction, most people don't look at this side of the active site. But we found by doing a general search over the whole protein, there's something very exciting and interesting about this pocket that hasn't been pointed out before. So that's very cool, and I got the opportunity, along with my co-authors Nicole and Marcel, to publish this data, and it felt really nice to be able to contribute to this broader conversation of redshifting, especially given that this was my very first research project. And so, after wrapping up any project, the next step is to consider the future directions on that project. So naturally, after wrapping up my first summer of research, I had some small future directions of my own to consider, such as, who am I, what am I doing with my life, and how do I get there? And as any good scientist knows, asking questions hardly ever gives you nice answers, but rather more questions. So this is a very overwhelming point in my life where I had to start thinking really seriously about my future. And when I tried to follow these lines of self-questioning, I just kept coming up blank. I knew that I really liked doing research, especially after that summer of doing it, and that I wanted to pursue a career along those lines, but that can look 100 different ways, and I had no idea where to start. So, inspired perhaps by my friend Firefly Luciferase, I thought that maybe I didn't need to know exactly what I wanted to do after college in order to just keep doing research, and that perhaps the answers would be revealed to me along the way. And so I opted for what someone making a flawed and strained metaphor might call a high-throughput approach, where I decided that perhaps focusing on my desired outcome, which is some kind of career in research, would end up addressing all of these questions all at once. I learned especially from that last piece of evidence I showed you on the Luciferase project that sometimes not knowing exactly what you're looking for helps you find something you wouldn't have found otherwise. And so I opted to just keep on doing research in different situations and see what I would find out about myself along the way. And that is how I ended up at the University of Illinois, Urbana-Champaign. As a fun aside here, if it wasn't already intimidating enough that I was going to a city I'd never been to a very scary chemistry program and department, and doing science I'd never done before, I also broke my brace the day before I started. So just please keep in mind that everything you see on the next few slides was done in a hot pink cast and an extra large glove. So, anyways. And at the University of Illinois, I worked for Scott Silverman, who studies DNA enzymes. That's catalytic DNA molecules. And the reason this is a cool thing to study is because most people don't realize that proteins are not the only catalytic biopolymers. So protein enzymes, like Firefly Luciferase and thousands of others, are ubiquitous in nature. They keep life afloat. That's the reason everything exists. So everyone knows about protein enzymes. RNA enzymes, or ribozymes, also exist in nature, and they have been sort of recently shown to have a lot of interesting and important biological applications in the body and also just in labs. However, DNA enzymes do not exist in nature, and so people didn't think they were really capable of catalysis, but given the obvious similarities between RNA and DNA, it makes perfect sense that DNA should also be catalytic, and in fact it is. The reason we care about RNA and DNA catalysts is because they can be generated de novo, or completely from scratch. And this is a really important property when you're trying to catalyze a brand new class of reaction for which an enzyme does not already exist. In that case, it's pretty easy to select an RNA or a DNA catalyst, whereas starting with a protein or creating a new protein altogether is very, very difficult. Now, engineering novel functions into proteins is indeed possible through strategies such as direct evolution or computational enzyme design, but these are very difficult processes, they're very time-consuming, and they often require some kind of known starting point already, so it's not an entirely de novo process. Now, I'm going to gloss over this, which kills the protein engineer in me. I would love to talk about this, but for the sake of this presentation and time, just know that these things are possible, but they're not always practical. Which is why DNA and RNA catalysts are sometimes the preferable option. So with DNA and RNA, people often think of RNA as being the superior catalytic biopolymer, but that's not necessarily true. Because RNA enzymes are found in nature and DNA enzymes are not, people often assume that RNA is just inherently more practical for these purposes. But when you look at the chemical and practical properties of the two biopolymers side by side, there are actually a few advantages to DNA that make it perhaps a better biopolymer to work with when we're talking about catalysts. For example, DNA can be directly amplified by PCR, whereas RNA requires additional steps. That just means that working with RNA is going to take longer because there's more steps we have to go through, whereas DNA, we can make a lot of it really quickly. DNA also has greater conformational freedom because it does not have a 2' hydroxyl group on the sugar backbone, which means it's more flexible, and that could be advantageous for catalyzing new reactions. It's also very easily made by solid phase synthesis, whereas RNA takes longer. It's got more protection and deprotection steps due to that 2' hydroxyl group, which means we can make it really easily in the lab, or we can just buy it. It's a lot cheaper. The other thing about DNA is that it's more stable at room temperature. So So all of these things make DNA way easier to work with than RNA. And on top of that, there is no evidence to suggest, with any hard truth that RNA has more catalytic potential. So for these reasons, it's totally worth studying DNA enzymes and seeing what kinds of reactions they can catalyze. And that's what the Silverman group does in Illinois. To identify DNA enzymes, we do in vitro selection. This approach applies iterated rounds of selection via enrichment and amplification. So if you imagine all those gray dots in the corner there are random DNA sequences, and the red dot is a catalytic DNA sequence or a DNA enzyme. We're first going to run a selection to enrich the more catalytic sequences out of the uncatalytic sequences. We'll amplify that pool, and then we're going to repeat this process over and over again until that desired catalytic activity, or the DNA enzyme, dominates the pool of DNA sequences. So if you recall that, similarly to the high throughput approach we use to find redshifted luciferases, this is also an outcome-driven approach. In both cases, we're going to end up with a pool of successful candidates without necessarily knowing how we got there. But also in both cases, we can then work backwards to analyze the sequences of our successful catalysts and look for patterns. And that's where we find interesting insights into mechanisms and such. So the Silverman lab finds DNA enzymes that catalyze a wide variety of reactions, but the class that I worked on was reductive amination, which is this reaction shown here. The reason we care about reductive amination is because the Silverman group recognized the chemical potential of exocyclic primary amines that are on the C, G, and A nucleotides of DNA as an opportunity for N-alkylation by reductive amination, which could be a helpful process for, say, site-specific labeling or maybe base editing, or just generally a helpful reaction to be able to catalyze in the research lab. And so in order to identify DNA enzymes that catalyze reductive amination, we set up an in vitro selection that looks like this, where we have the two reaction partners, which is one of these C, G, or A DNA substrates, and some kind of aldehyde on the other side. We attach those two reaction partners to short DNA strands that are complementary to two binding arms on either end of an N40 region. An N40 region just means we have 40 nucleotides long of DNA that's totally random. And this is what may or may not be a DNA enzyme, based on whether or not it helps catalyze the reaction shown in green. This is a very clever selection design because it makes it super easy to differentiate your catalysts versus your non-catalytic sequences on a gel. So if down here we have the uncatalyzed starting materials where this reaction did not occur, we can see that when the reaction does occur, we get a mass shift from the additional mass of the oligo attached to the aldehyde, so that we have two distinct bands on a polyacrylamide gel. So that's really easy. You can just pull out everything from the top gel and re-select on that and ignore everything in the bottom gel. So following this process, the Silverman lab did successfully run this selection using a benzaldehyde reaction partner, and they identified like 40 new DNA enzymes, which was really exciting. And when I got there, they wanted me to test the viability of a similar but slightly different reaction, swapping out the aromatic aldehyde for an aliphatic aldehyde, hypothesizing that perhaps this aldehyde would be more reactive. And so in order to test the viability of this selection, I had to build this aliphatic aldehyde reaction partner, which just meant connecting that aldehyde onto a DNA strand. And the short version of this process is that I pursued two different syntheses, and this one worked and that one didn't. The long version of this process is that it took 7 weeks and was very frustrating, and that's not very exciting to talk about, so I'm not going to. But anyways, I built this aldehyde down here attached to DNA, and then I got to use it. Unfortunately, since that took so long, I didn't get to run the actual in vitro selection of DNA enzymes, but I did get to do arguably the most important part, which is to determine the background reactivity of the two substrates. So the reason we care about this is that if we were to put these two reaction partners in solution by themselves without a DNA enzyme, and they were to react on their own, then there's no point in finding DNA enzymes in the first place if the reaction's going to happen spontaneously. So the first thing we need to do is make sure they're not doing that in excess so that we can ensure that we'll have good rate enhancement when we find DNA enzymes later. So this is what that selection looks like. It's pretty much the same as the one before, except for instead of that random N40 region, these two things are just connected directly together by a splint. And this is what the results of that looked like. I'm going to explain that better. So down here, this is the uncatalyzed starting materials. This is where no reaction has happened. And then when those two things did spontaneously react, we see a mass shift up here. There's this faint product line up here over 96 hours at 2 different pHs. And this is just one of the substrates. This is the triple G substrate. There was also A and C, but it's a lot of gels, so I didn't want to show all of it. But you can see that, importantly, although there is product, the yield is very, very low. It's less than 5% across the board with all of the substrates. And that's good. That means that background reactivity is sufficiently low in order to continue with in vitro selection of DNA enzymes, which is good. That means that they can continue with this project that I worked very hard on. The other thing I did was compare the reactivity of the aliphatic aldehyde with the benzaldehyde that had been used in previous selections, and I found that the reaction kinetics of product formation was relatively similar across the board, which is a little surprising because we thought maybe there would be a slight reactivity difference. But really this is a good thing because the benzaldehyde results of the DNA enzyme selection using the benzaldehyde went really well, and so we can ponder, perhaps, that if the reaction kinetics of that one ended up being favorable for DNA enzyme selection, then we might have a similarly successful selection with the aliphatic aldehyde. So this was really cool. This is where I wrapped up my summer at Illinois and left once again to consider my future directions. So at the end of this summer, I realized that I still did not know the answers to any of these questions. However, I had found that in choosing to ignore these questions and by chasing outcomes rather than testing hypotheses, I ended up learning a lot about myself. And what I had learned about myself was more than enough for me to decide that I wanted to continue studying chemical biology in a PhD program. And so I applied to programs, I toured schools, and I chose one. And today I am 2 weeks away from starting my PhD. And I still don't know the answers to any of those questions. But I have learned through all my years of research that I don't necessarily need to know the answers to those questions, because what I do know about myself and what I will continue to learn about myself will continue to be enough to keep pushing me in the right direction. And so I am very excited to start my PhD, and I'm really glad that I've learned all this stuff about myself because it means I know exactly what to look for in a research project, in a lab, in a mentorship style, etc. But I still don't know exactly what I'm going to do after I get my PhD. But the great thing about getting a PhD, perhaps the best thing, is that I will have 6 very long years to figure that out. And with that, I'd like to thank my mentors, Aaron LeConte here at Keck, as well as Scott Silverman at the University of Illinois, Urbana-Champaign. I'd like to thank the group members that helped me in all of these projects, especially the ones that showed up today, as well as my various sources of funding, and of course, Scripps Presents for having me. This has been a really exciting experience for me, so thank you very much.